So what we're going to do now is go over how organisms get arranged into different groups. One of the actual tools, or strategies of doing that for scientists, and it's one of the tools that we use are called cladistics. Right. Now, the people who do this perform a type of science called systematics. Which has to do with both naming and classifying. And there have to be a variety of different tools that are used in doing this. And so some of them are developmental, some of them are molecular, some of them are physiological, and for different organisms, some of things might be used more so than others as well. So cladistics are one way of organizing and splitting off groups of organisms to look at their relationships and kind of see who is maybe more closely related to one another or more distantly related to one another. And so the example I'm going to use here is going to be a loose example for the phylum Chordata. Okay, so the chordates. So a cladogram is a diagram. It's a branching diagram. So the cladogram is a branching diagram. And that's what I'm going to be drawing here as a cladogram. And cladistics are some of the rules and things that are applied to this and some of the terminology. So we have to have a common ancestor or some common ancestral trait that all the organisms in this cladogram are going to share, right? If they don't have a common ancestor or a common ancestral characteristic, then you're already starting off in the wrong place. You have to have something that's common for all of them that you have to list. And so for chordates, there are a whole number of different features, which you'll study later, like a notochord and a hollow dorsal nerve cord. So they're all things that all members of the group have to have at some point in time in their life. And if they don't, then they're not part of that group. That's kind of the idea. So there has to be something that we point to as they all share this. Then what happens, so now all the members are part of this group. Now we come to what's called a node. So the node is, the way to think about it, is a decision point. And that's what really the cladogram is, sort of like a decision matrix, yes, no, for certain particular features. This particular node here, we're going to have our first node, we're going to say, do they have vertebra or not? And so anyone following this line here from this point forward is going to be yes, they all have a vertebrae. Anyone who doesn't have a vertebrae, no, is going to then get pulled out, right? So we have, for example, our cephalochordata like an organism called Amphioxus. Amphioxus has all the common chordate characteristics, but it doesn't have vertebrae, and it never develops a vertebrae. It just keeps the notochord and hollow dorsal nerve cord, and that's it. It doesn't develop anything else. So it gets pulled out as its own group, and the rest of them kind of continue along this path. So now we have all vertebrates. So for the vertebrates, how do we divide them up? Again, people do choose what characteristics to put next. And so that's where there's a lot of revision that goes on, because a group or team of people will choose certain characteristics to come next, and then someone else will say, I did the same work and I chose a different characteristic to put as the next one. Why did you choose the one that you did? And so this is one of the things. In addition to that, a cladogram, each node should be of the same type. So if you're doing a cladogram that has morphological traits, they should all be morphological traits. If you're doing a cladogram with physiological traits, they should all be physiological traits. If you're doing a cladogram with genetic sequencing, then each of these branching points should be a specific variation in gene sequence. You should not be mixing them up. And what you can do then is, we'll get to a little bit, is take multiple cladograms, each one representing different aspects of or different ways of organizing a group of organisms, and then compare them and then say how do they agree with one another, do they disagree with one another, and then eventually come up with relationships from that. So it's a very big process. In this example, though, what we have, first node, vertebra, second node, we're going to have jaws. So a hinged jaw. And in this particular case, we have Agnatha, the jawless fish. They would come out, and everybody else who has vertebrae and jaws would stay in the group. We could then have another branching point. So now we have all the organisms, all the vertebrates that have jaws. So then another way to separate them out. Well, we have fishes and amphibians and birds and reptiles and mammals and all these groups. So one of the things we could pick next are forelimbs. So now other groups of fishes, so the jawless fish here, the osteichthyes, the bony fish, and the cartilaginous fish. So I'm just going to simplify it and just put other fishes. They come out here, and other organisms stay. So each point here is each another node. You know, this node here may be amniotic egg. So amphibians lay eggs, but it's not an amniotic egg in that there's not a shell. It does not have these certain layers and compartments. One with amniotic fluid called an amniotic egg. Birds lay this type of egg. Reptiles lay this type of egg. And there are mammals who lay eggs that also have it, but mammals also still have an amniotic sac, an amniotic fluid that all the organisms develop in. Even though not all of them lay eggs, they still have this amniotic structure. So they would all fall along this line, but the amphibians would not. The amphibians are vertebrates with jaws and 4 limbs, but they don't have amniotic eggs. So they would get pulled out. Still have all these other groups. Okay, so how do we separate them? So we could then go here and say, so after the amphibia, we have all the organisms with amniotic eggs. You know, how do we potentially pull them out? Well, in this particular case, we could say hair. Okay, and so now we have our reptiles here, which is again we'll talk about that in a second. And then the mammals will go up here. And so this is a simple one in that there should be and can be a lot more characteristics put in here, but these are all chosen characteristics, and that any organism along this path would have them. So mammals have hair, an amniotic egg, 4 limbs, jaws, they're vertebrates, and they should have all the chordate characteristics. And then as we go along these side branches, we would say, okay, the mammals have this derived trait that these others don't share, that they have hair, for example, but they do share these ancestral traits. Okay, so we have some traits that are ancestral, these shared ones, and then we have derived traits that are unique. That's kind of how we pull them out and separate them. So this group, if we looked at this whole big group, would be considered we call a monophyletic group. So mono meaning one. So we can trace back, or we can link all these organisms through a common ancestor with common ancestral characteristics. Right. Reptiles, for example. Now we see where are the birds, right? Well, that's because birds are essentially sort of put in here with the reptiles for a number of reasons, and the group that you might think of as reptiles isn't really, when following this system, a cohesive monophyletic group. It is really a paraphyletic group, meaning that what would happen is we have a cladogram, let's say like this, and we just have organisms here, A, B, C, and D, like that. And so one of the things a cladogram could be used for is just looking at, who's more closely related. So just by looking at this drawing here, you would say, okay, well, C and D, whoever those organisms are, groups of organisms, they're more closely related to one another than, for example, D is to B or D is to A, and so forth. So then you can say, okay, so that this group, B, C, D, they're more closely related to one another than, than to A, and so forth, because they have this shared point here that they all have along that particular branch that A doesn't, that pulls them off. Now what we have, especially when we look at a group that we call reptiles, is that we'll find some members like this being pulled out into a class, and then another group that's sort of technically, because of all these characteristics, more closely related, but isn't included in that group. We bring it out and then say it has its own separate group or separate class. And so the cladogram says one thing, but our way of nomenclature and talking about the organisms doesn't, it's not that it doesn't agree with it, it's just we almost ignore it and we just use something totally different. So in that particular case, this is paraphyletic, meaning that, say, here, all members of the group are not included. I mean, some members in the group are not there. They should be there, but they're pulled out to their own separate group for some other reason that doesn't agree with the cladogram. And so those are things that we'll talk about a little bit more in detail with as we talk about some of the groups. When we talk about reptiles specifically, talk about very specific details of the ideas of them being paraphyletic and why and what characteristics they share, what characteristics they don't, that make some of the groups unique. But this is the idea. So cladistics is one of the tools used in classifying organisms, used in systematics. And then one of the next things we talk about is phylogeny. Phylogeny is what then looks at evolutionary relationships and will start to include evolutionary time. So our timeline that we had talked about sort of put these things in place, because the distances drawn here aren't necessarily timeline, representative. It's just to keep things organized. The phylogeny, or what's called phylogenetic systematics, starts to look at the evolutionary relationships, including information from cladistics and fossil evidence from evolutionary timelines. So the phylogeny, which is really evolutionary relationships, includes a lot of information, and that builds something called a phylogenetic tree, which is then far more complex. It might use multiple cladograms, like I said, and other information to put it together. So the cladogram is just a more contained, very specific thing that has certain rules that are followed, and you should just be familiar with this.